السلام عليكم يا دكاترة. النهاردة ان شاء الله بإذن الله هنخلص الجرام نيجاتيف روتس، وكده هنكون خلصنا الجرام نيجاتيف روتس كلها. هيتبقى لنا جرام نيجاتيف كوكاي، ودي اللي هي النستيريا، وغالبا الدكتور علاء دايما بيحب يشرحها لكم. طيب تعالوا بقى نبدأ النهاردة مع مجموعة مختلفة شوية خالص. هي هتلاقوها انك في الشكل بتاعها مختلف، احتياجاتها للريتشد ميديا بتاعتها والنزرعها والنيوترنت مختلفة تماما. نبدأ بقى اول واحدة اللي هي الهيموفيلس انفلونزا. اهي بيقول لحضراتكم الهيموفيلس سبيشيز. اهم حاجة في الموضوع طبعا ان احنا مكتوب قدامنا بيليومورفيك، يعني شكلها غير محدد، ما احناش عارفين نقول انها رودز ولا كوكو رودز ولا كوكو باسيلاي. الشكل قدامكم اهو في السلايدز واضح جدا. كمان في ميزه مهمه جدا ان هي كابسوليتد، من الحاجات القليله جدا الانكابسوليتد بايوجينز كمان. عشان كده للاسف دي من اشهر الانواع اللي بتعمل مننجايتس، يعني التهاب سعائي عند الاطفال، هي الهيموفيلس انفلونزا اللي قدام حضراتكم. بنقول عليها كوكو باسيلاي رودز عشان الشكل اللي انتوا شايفينه دي. بنقسمها سيروتايبنج ل6 انواع من الاي للاف، اشهرهم واهمهم اللي هو تايب بي اللي بيعمل المننجايتس. طيب في السلايد بيقول لنا محتاجه حاجه مهمه قوي قوي عشان تتزرع عليها. احنا بنزرعها على شوكليت بلاد اجار. هي عايزه اكس فاكتور اللي هو الهيمين. قدام حضراتكم اهي محتاجه الاكس فاكتور اللي هو الهيم، ومحتاجه كمان فاكتور في او النات اللي هو عباره عن نيكوتين ادونين داينيوكليوتايد. الاثنين دول محتاجيهم عشان تنمو على الميديا. يبقى هو شوكليت بلاد اجار لازم يكون فيها اكس فاكتور والفي فاكتورز. هي الوحيده اللي بتنمو على الميديا دي، فتبقى سبيسيفايد لها وسلكشن، فتبقى دي هيموفيلس انفلونزا. تعالوا نشوف بقى ايه عوامل الشراسه بتاعتها وايه اللي بتخليها تعمل مننجايتس. تعالوا بقى نشوف دلوقتي بقى الفيرولنس فاكتورز فور هيموفيلس انفلونزا. فيرست كبسول encapsulated IgA proteases. بروتين degrades IgA الانتيبوديز، اللي هو الانتيبوديز المفروض بيحمينا. هي عندها بروتين بتفرزه اسمه IgA proteases يحلل لنا IgA بتاعنا فتخسر جهاز المناعة. يبقى ده facilitating attachment to the respiratory mucosa. Then after that it becomes established in the upper respiratory tract. The organism enter the bloodstream. Now it's bacteremia and spread to meningitis. Also she has lipid A and pylori. All of these are virulence factors help her to be virulent. Okay. Lab diagnosis: a specimen from sputum or cerebrospinal fluid CSF. The stent will be gram-negative encapsulated coccobacilli. As we said before, it's polymorphic. Culture على chocolate blood agar containing X factor with V factor, as we said before. Treatment: drug of choice for meningitis or other systemic infection caused by Haemophilus influenza is cefotraxone. It is the drug of choice. Amoxicillin and clavulanic acid combination can be used. Ampicillin also can be used. And the most important, we have vaccine for Haemophilus influenza that make it not spread because it has vaccination. Now we will start another group of gram-negative. It is called curved root. Curved. We have 3 important species: Vibrio species, Campylobacter species, Helicobacter. We will study first the Vibrio species. طيب نبدأ بالفيبريو species. We will start with Vibrio species. The most important is Vibrio cholera. The major pathogen in this genus is Vibrio cholera, which is caused cholera. How can we infect by cholera? By eating raw or uncooked seafood. Vibrio cholera can be transmitted through food. The most important property for Vibrio species, they are curved. Now we will start with curved or comma shape. This is diagnostic under the microscope. Gram-negative roots, non-spore, highly motile with single polar flagella. We divided Vibrio cholera into two groups according to nature of its O cell antigen. Members of O1 group cause epidemic disease. What's the meaning of epidemic? You will know in public health. Where non-O1 organism causes product disease or non-pathogen disease. Most characteristic for Vibrio cholera, it loves salt water. It is halophilic, require high percentage or concentration of sodium chloride to live. It is oxidase positive, facultative anaerobic, tolerate alkaline. This is the most important character for Vibrio. So Vibrio cholera is transmitted by fecal contamination of water and food, primarily from human resource. Human carriers are frequently asymptomatic with no symptoms and also include individuals who are either in incubation period or convalescent فترة الحضانة. Spread by contamination of water or through animal marine fish, shrimp or oyster. And the most diagnostic characteristic for cholera is rice water stool like rice. The pathogenesis and how we know the patient is infected with cholera. The pathogenesis depend on colonization, where it must be in small intestine, and secretion of enterotoxin. And for colonization to occur, it must be large number of bacteria must be ingested, because the organism is sensitive to stomach acid. We said it is alkaline. So the patient who takes antacid or who have had gastroectomy are much more susceptible to cholera. Once adherence to the cell of the gut, which is requirement for colonization, so the bacteria will secrete a lot of enzyme mucinase. Mucinase enzyme dissolve the protective glycoprotein coating of the intestinal cell. After adhering, the organism multiply and secrete another toxin called enterotoxin chloragine or chlorotoxin. It is specific for Vibrio cholera, and this is the main cause of loss of electrolyte. So as we said, this toxin chloragine is the main cause of severe fluid and electrolyte loss. Now we need to make laboratory diagnosis. Laboratory diagnosis, the specimen will be taken from rectal swab or watery stool, and the culture it will be on alkaline peptone water. As we said, it loves alkaline or thiosulfate. For treatment, the first step, it must be rapid replacement of water and electrolytes. This is the first treatment. Then use tetracycline. Why cholera is now not spread like before? Because now we have two important vaccines. Two are oral vaccine. One called Dukoral contained killed whole cell of all strains. This with recombinant cholera toxin subunit B antibodies, which induced by vaccine prevent the ingestion of the Vibrio from attaching to intestinal mucosa, so prevent the first step of colonization and neutralize any cholera toxin that is produced. Second vaccine is killed whole cell vaccine called Chanchol, contains both O1 and O159 strain. Okay, so we have two vaccines for cholera. Second species is Campylobacter species in the curved root. The important properties of this species are curved gram-negative roots that appear either comma or S-shaped, as we can see in the figure in front of you. They are microaerophilic. Microaerophilic means they grow best in 5% of oxygen, only 5%, not 20% that found in the atmosphere. We will know how we have Campylobacter infection. How we can infect through Campylobacter infection. Domestic animals such as cattle, chicken, and dogs serve as a source of the organism for human. Transmission is through fecal oral. Food and water contaminated with animals' feces are the major source of human infection. Foods such as poultry, meats, and unpasteurized milk are commonly involved. Also, puppies with diarrhea are a common source for children. Human-to-human transmission occurs, but it's less frequent than animals-to-human transmission. Pathogenicity of Campylobacter species. Enterocolitis is the first clinical finding. Begins as watery, foul-smelling diarrhea followed by bloody stools accompanied by fever and severe abdominal pain. This is the most characteristic for a pathogenesis of Campylobacter. We have two types of Campylobacter, or two species, Campylobacter jejuni, and this is sensitive to naldexic acid and grow best at 42 degrees. And this is the cause, or the main cause, of gastrointestinal. Campylobacter fetus is resistant to naldexic acid and grow best at 25, and this causes febrile infection or mild infection. Laboratory diagnosis: we take a specimen from stool and we grow on blood agar containing antibiotic. A biochemical test is oxidase and catalase positive, but urine negative. Treatment: erythromycin and tetracycline. Now, the third species is Helicobacter species. The main cause of gastritis and peptic ulcer is Helicobacter pylori. Infection with Helicobacter pylori is a risk factor for gastric carcinoma. What is the most features or important feature for Helicobacter? They are curved gram-negative roots similar to Campylobacter. Campylobacter, we said it is urease negative, and Helicobacter pylori is urease positive. This is the main difference between them. Acquired by ingestion of food, we can take a Helicobacter pylori. Pathogenesis and epidemiology of Helicobacter pylori. First, Helicobacter pylori attached to mucus secreting cell of gastric mucosa. Then it will produce large amount of ammonia from urea by urease enzyme. This will be followed by inflammatory response, lead to damage of mucosa. Also, loss of protective mucus coating predisposed to gastritis and peptic ulcer. And ammonia also neutralizes the stomach acid, so allowing the organism to survive more and more in the stomach. Laboratory diagnosis: the organism can be seen on gram-stained smear of biopsy specimen of the gastric mucosa. Also, it can be cultured on the same media as Campylobacter. In contrast to Campylobacter, Helicobacter pylori is urease positive. Urease production is the basis for non-invasive diagnostic test called the urea breath test for detection of Helicobacter pylori. In this test, radiolabeled urea is ingested. If the organism is pressed, urease will cleave the ingested urea. Radiolabeled carbon dioxide is evolved and the radioactivity is detected in the breath. This is brief account of the breath test. Also, a test for Helicobacter antigen in the stool can be used for diagnosis and confirmation test for Helicobacter infection. For treatment, we must use antibiotic. First of all, combination of two antibiotics is used to decrease resistance. We use amoxicillin and metronidazole with bismuth salt to reduce the gastric acidity. Tetracycline can be used instead of amoxicillin if resistance happens. There is no vaccine or other specific preventive measure to prevent Helicobacter pylori.